So, Melissa, good morning. Welcome
Good
to
morning.
welcome to India.
Good morning from Idaho.
Yeah. So we are so excited today to witness some marvelous things from your classroom.
Well, I've got a couple of things to show you guys today, so hopefully they'll be interesting.
Yeah, definitely. My students will join very shortly, maybe within a few minutes. They are going. Yes, they are right now. They are joining one by one. Um,
Excellent.
Melissa, if you don't mind, would you like to show your classroom a bit so that we can see how your classroom looks like, how different your classroom from our classrooms?
Yeah, that's actually where I wanted to start today because I want to introduce you to a couple of my friends. I have some class animals that live in my room, and my teaching partner has one in her room too. So I was going to do a quick tour of the classroom. I've got my cell phone here, so I'm going to join on my cell phone and walk around the room showing you things, and then I'll come back and we can do a couple of demonstrations.
Yeah, sure. Yeah. So how is the weather at the moment in your country, in your region? How is the weather like?
That's an interesting question. Last week, so we use Fahrenheit in the United States, which is very awkward for talking to people from the rest of the world. But last week in this area, it was about 55 degrees Fahrenheit, which is pretty warm for this time of the year. And then we woke up this morning to 3 inches of snow, and we're supposed to get hit with a big snowstorm again today and tomorrow. So by Sunday, we should have probably 6 inches of snow outside, and it's about 30 degrees Fahrenheit. So it's been kind of a crazy week. Temperatures started high and they're ending low. So
Yeah, that's really interesting because India is actually the place, the region where I am living is quite tropical weather conditions. And it's always hot and humid and warm. So we never ever experience the snow conditions. If we see the regions where you live, I think we see quite unusual because the weather you experience definitely is different from the weather we experience. So let us not waste our time. Please show your classroom. We are so excited.
yeah, maybe I can step outside for a second and show you guys the snow too.
Yes. In the meantime, I think we are back to normalcy in my country, and no more masks and no more sanitizers. Everything is
Okay.
quite normal. And the interesting thing that we have to share with you is that in India we have developed the healthy immunity, and we are all actually are right now safe from the COVID-19. We have no more threat from the COVID-19 because everything is just normal, and the vaccination process has kick-started, but very few are actually willing to take the vaccination because people are not taking the vaccine anymore because we have already developed the immunity. So people are not getting the vaccination, and the vaccination started, and the vaccination is in progress, but it is only the frontline people like the health assistants and the doctors and teachers only taking the vaccination, but not the common people. But the common people are not really worrying about the vaccination.
That's really interesting. Yeah, we're still in masks. I have my mask right here. The only reason I'm not wearing it is because there's nobody else in my room right now but me. We are still doing sanitizing. We're still only going to school 2 days a week, so I only have half of my students here. But we are working on the vaccine. I get my second dose of the vaccine tomorrow. I got the Moderna version of the vaccine, so it's an mRNA virus vaccine. And you have to get 2 doses about 4 weeks apart. So I'm getting my second dose tomorrow. Right now, in most of our states, just frontline workers, healthcare workers, and teachers are getting the vaccine. They just started approving it for people older than 65 years. And we don't really expect that the general public will even be able to get it for a couple of more months. But it's a weird mix here of some people are really excited about the vaccine and can't wait to get it, and other people are afraid of the vaccine. They don't understand the science behind the vaccine and it makes them nervous. And so they're choosing not to get the vaccine at this point. So it's interesting.
So Melissa, shall I allow my students to ask you a few questions?
Yeah, definitely. I'm going to go ahead and jump into this other. There we go. Can you hear me okay? I'm jumping into my other.
I can hear you. I can hear you. Yeah. So Sushmita, do you have any question? I would like to start with you, Sushmita. So do you have any question? Bhagyasri, do you like to ask a question?
Yes, sir.
You can greet our teacher, Melissa, and fellow. So you can ask Bhagyasri. Our teacher is there, and she is showing her classroom. Just like look at the classroom. How is Yeah, all of you are requested to look at their classroom, and this is the classroom setup in United States of America, and it looks like a museum. And in fact, in our classrooms are different from the United States of American classrooms. This look is quite amazing. Everything is there. I think students really enjoy sitting and seeing these images, these things, and everything is there. And so they are so lucky to have these things, and they are so lucky to have this great teacher. So Bhagyasri, you please ask the question. Yes, look at that one. I think it looks like a bow, I think. So Melissa, could you please explain about it? Yeah, she's going to show us, and everybody should look at that. Yeah. Melissa, if you don't mind, could you please explain about it? So Melissa, we can't hear you. We can't hear you.
There, I'm trying to run two devices and the audio is not being super friendly on one of them. So this is my corn snake. He's about 12 years old. He's lived in my classroom for about the last 12 years. His name is Guttata, which is his genus name. It means speckled or spotted. Corn snakes come in a lot of different colors, but one thing that they all have in common is this speckly spotted pattern that helps them hide and live in fields. They tend to like to live near people, so they're not very aggressive. They're a pretty calm snake because they like to live in agricultural fields where people are growing crops, because that's where they can find small mice and other small mammals to be able to eat for food. He's got a really pretty kind of pinkish color here. He is not a true albino, though if you look carefully, you can see he has red eyes. He's what's called hypomelanic. So he doesn't have the same amount of melanin as a regular corn snake, but he definitely has some because he has some body color still. Does anybody have any questions about my friend Guttata?
Yeah. Bhagyasri, do you have any question? Please ask the question. Bhagyasri.
Yes, sir.
Yeah, go ahead, Bhagyasri. Ask the question.
Yes, sir. Hi, ma'am. My name is Bhagyasri. I am studying 7th grade. My hobbies are reading books, playing games. My question is, what is the best method to teach biology?
What is the best? I missed the last word. What is the best what?
To teach biology.
Oh, why do I like to teach biology?
Oh,
Yes, ma'am.
that's not the question.
Oh, that's an excellent question. Oh, that's not her question?
No, that's not our question. Actually, our question is, what is the best method? What is the best method to teach biology? What is
Oh, the best method to teach biology.
That's the question.
Well, I think it's really important that students are able to actually touch the things that they're studying and actually do experiments and actually, in my area, we call it place-based learning, which means that the students actually go outside of the classroom. And one of my favorite things to do with my students every year is we do a lot of water quality testing. So we look at how much nitrogen is in the water and how much dirt and silt is in the water and how that affects the plants and animals that are living there. So when we study the plants and animals, we actually go out into the local lake, out into the local marshlands, and we actually study things by looking at the things around us. I think that's the best way to understand biology, is to actually do biology and be outside of your classroom.
That's excellent. That's excellent explanation, Melissa. Melissa, I have a question. What kind of food actually you offered for the snake? Do you have any specified food that this particular animal, this creature feeds on?
That's a good question. So my snake only eats about every 3 weeks. Can you imagine going 3 weeks without eating? His body doesn't use food in the same way that our bodies use food. You may have heard the terms cold-blooded and warm-blooded, and they refer to how an animal gets their energy. Humans are warm-blooded. We have to eat food in order to make most of the heat energy to keep our bodies warm. But snakes, like other reptiles, they get a lot of their heat energy from the world around them. So you may have seen a snake laying on a warm rock. You may have seen a snake laying on a warm road because they're getting some of their energy from the sun and from their environment. So my snake friend here only eats about every 3 weeks. And what he eats are frozen mice. Now, some snakes won't eat the frozen mice. They will only eat live mice, which is a little bit dangerous because when you're trying to feed your pet snake a live mouse, that live mouse will fight back. They'll scratch the snake. They'll cause infections to the snake's face. So it's actually kind of dangerous for the snake to eat a live mouse. But my snake is really good about eating a frozen mouse. They come in little packages, and he eats about 3 frozen mice about every 3 weeks. Now, he is a male snake. He's a boy. And about March of every year, he stops eating for a while. He dedicates all of his energy and all of his interest to trying to find a mate. So he goes looking for a girl snake. So he doesn't usually eat at all from March until July. He goes from March until July without eating at all because he's trying to find himself a girlfriend. He's not going to find a girlfriend. There are no girl snakes in my classroom, but he doesn't know that. He keeps trying.
That's very interesting information, Melissa. Melissa, other than this particular snake, do you have any other features in your classroom?
Yeah, I'm going to go ahead and carry my Chromebook because my cell phone is not being super cooperative here. Will you put him away for me, please? Just put him away for me, please. He's wrapped around my... I know, just put him away. So the next friends that I have here with me today are my turtles. Actually, no, I'll just take one out of the tank here. That'll be the easiest way to see them. So this is my friend, Juan. Juan is a female red-eared slider turtle. And then Juan's friend is Beaker. Beaker is what's called a Western painted turtle. If you look at her underside, you can see she has this really pretty red and yellow coloring underneath her body that makes her look like somebody painted her. So those are my 2 turtles, Juan and Beaker.
Very interesting.
And then I'm going to walk you next door and we'll see if I can get a good picture of, good morning. This is my friend's frog.
Oh,
Her name is Goopy. What kind? She's a White's tree frog? She's a White's tree frog. And she likes to eat crickets. And she likes to stare at students. And it almost looks like she's smiling at them. She's looking at me right now like, why are you staring at me? Why are you looking at me? Good morning, Goopy.
hi, good morning from India.
There, Goopy just made some friends in India this morning.
Navya Sai, do you have any question? I start with you, Navya.
Hello, ma'am.
Good morning.
Good morning, ma'am. My name is Navya. I'm studying 10th grade. Can I ask a question?
Oh, definitely. I love questions.
How do you write a research question for an experiment?
Ooh, that's a good one. So, first of all, I think it's really important to find something that you're interested in. I think that it's much easier to do research if you find a topic that you're interested in. And then when you start writing the research question, one thing I always tell my students is make sure it's not a yes or no question. We don't want a question that you can just easily answer with yes or no. So thinking about a question that's going to allow you to come up with a better answer than just a yes or no is going to be an important next step in writing that research question. Are you guys working on some research projects right now? That sounds interesting.
Taslima.
Thank you.
Taslima, I didn't-
Hi, ma'am.
Good morning.
Good morning, ma'am. My name is Taslima. I am studying 7th grade. My hobbies are reading books, playing games. Can I ask you one question, ma'am?
Definitely.
Do your students enjoy your class?
I would like to think my students enjoyed my class. I'm probably one of the teachers that does the most hands-on projects, the most experiments. I also really like to take my students places outside of my classroom. I take my 8th graders to Yellowstone National Park every year, and we wear snowshoes, and we go hiking, and we do water tests on the different thermal features in Yellowstone, and we get to see the big bison, the buffalo that live in Yellowstone. I like to take my students to Florida every year, and again, we do water testing. We also go swimming with manatees, and we dissect sharks. Today in class, we're actually dissecting an elk heart to learn about the circulatory system and to have a happy Valentine's Day present. So I think that the students do like to be in my class because we do a lot of exploration and a lot of hands-on learning, not a lot of stuff out of a textbook.
Melissa, within
Thank you,
few
ma'am.
minutes, students, we are so lucky today to witness one experiments done by our great teacher Melissa, and she is going to do 1 or 2 experiments because I'm so excited to see the experiment, especially the strawberry and as well as the another experiment that ma'am has shared with me. And meanwhile, I would like to ask some of the questions, some of the students to ask some questions. Are there any students who can ask some questions quickly? Nahin, do you have any questions?
Yes, sir.
Nahin or
Hi, ma'am.
Good morning.
Good morning, ma'am. My name is Nahin. I'm studying 9th grade. My hobbies are reading books and playing games. Can I ask you one question?
Yeah, please.
What is your teaching philosophy?
So that kind of goes back, that's a great question. That kind of goes back to one that one of the other students asked me a few minutes ago. And I really think that it's important for students to get their hands on learning. I often tell people that in science, you have to touch it and smell it and taste it and roll around in it in order to really understand it. So to me, my philosophy of teaching is that students are active and doing science, not just reading about other people doing science. And then the other part of my teaching philosophy is that failure is really important. I know sometimes we tell people not to fail and say that failure is bad, but I think that we actually learn a lot from failure. In science and in engineering, people fail all the time. And it's because we fail that we learn new things. So in my class, I tell the students to fail forward. It's okay to fail as long as you don't fail and quit. You have to fail and learn something from it and move forward. So I tell my students all the time to fail forward.
Thank you, ma'am.
That's great. That's really a great inspirational thing that we have to learn. Yes, Neeraja, do you have any question?
Yes, sir. Hi,
Neeraja.
ma'am. Good morning.
Good morning.
My name is Neeraja. I am studying 9th grade. I want to ask you a question, ma'am.
Okay.
How do you teach biology for your students?
So how I teach biology for my students is to give them lots of opportunities to actually do the content. I actually want to go ahead and do an experiment for you right now. How many of you have already studied cells in biology? Have a lot of you already talked about cells?
I request to all the students to respond quickly. And how many of you have studied the topic cells? Taslima or Navia, have you studied the topic cells?
Yes,
Have you
ma'am, I studied.
So one student has
I
replied
also
that.
studied.
So how many of you have studied the topic cells in your biology textbook? So I think almost all of you have studied the cells part. Yes, Melissa, I think I have understood that all of these students have the topic named the cells in their textbook. So you
Okay.
can please go and show the experiment. We are going to enjoy it. Truly.
Excellent. Okay, so you all know that in cells, in most plant and animal cells, there's a nucleus in the center of the cell, and that inside that nucleus is the deoxyribonucleic acid, or DNA, that is the code for all of a living thing. It tells the living thing how to make proteins. It tells the living thing how to deal with waste products. It tells the living thing, it actually gives us all of our traits and our characteristics. So humans get 2 copies of every chromosome. We get 1 copy from our mom and we get 1 copy from our dad. That means we are diploidy, 2 copies. However, we have a fruit in the United States called a strawberry. I don't know if you guys get to see or eat strawberries very often, but this strawberry here is kind of weird because instead of getting 1 copy of the chromosomes from mom and 1 copy of the chromosomes from dad, strawberries can get up to 8 copies of the chromosome. They're what's called octoploidy. So they have lots and lots and lots of extra DNA inside of their cells. They have lots and lots of extra chromosomes inside their cells. So if I take this strawberry and I put it in a Ziploc bag, I can actually pull all of that DNA out of the nucleus. But there's a couple of steps I have to take along the way. The first step is I'm going to use my fingers this morning and I'm going to mash the strawberry up. If you guys remember studying cells in science, you remember that cells, especially plant cells, have a really thick cell wall around the outside that helps protect and keep the structure and shape of that cell. So I'm going to use my fingers right now, and I'm going to kind of mash up that cell wall. I'm just going to smash this strawberry with my hand and try and tear open that cell wall. Oops, I mashed a little too hard and I broke my bag. Then, once I get the cell walls nice and smashed up there, the next thing I want to do is I want to add a chemical to this strawberry mash that's going to help pull that DNA out of the smashed up nucleus and out of that smashed up cell wall. So I'm going to use a chemical that's called an extraction buffer. Now it sounds kind of fancy and kind of sciencey, but in reality, this extraction buffer only has 3 ingredients. It's made of water, soap, and salt. When you mix the water and the soap and the salt together, they have a chemical reaction with that strawberry that helps pull that DNA out of the nucleus. So I'm going to add a little bit of this extraction buffer here to my strawberry. And then again, I'm going to mix this around. Try and get some more of that strawberry down there into that buffer. I'm going to mix this around. So this is just soapy, salty water. At this point, my kids always want to eat it because they think it smells good. It smells like a strawberry milkshake, or it smells like a strawberry fruit drink. And I have to tell them, don't eat it. It's got soap and it's got salt in it. It's not going to taste very good. It's going to make your stomach upset. Then the next thing I want to do is, now I have all this extra pulp in here. I have all this extra cell wall. I have all this extra cell parts that I don't want. So the next thing I'm going to do is I'm going to run it through a strainer. So I'm going to take this funnel here and I'm going to add what's called cheesecloth. It's a really fine piece of mesh. It's got little holes in it, and I'm going to use it to strain some of that pulp out of my strawberry. So I'm going to pour this in the top here of my strainer. And hopefully you can see it starting to drip down there into the bottom. Kind of squeeze a little bit here on the top so we can get a little more liquid down there. Trying to get rid of all those extra pieces of the strawberry that I didn't really want. You can see the liquid starting to kind of fill up in there. There goes a big chunk of it. Okay. So at this point, I'm going to get rid of this mess. This is all of the other cell parts that I didn't want. And now I've basically got my DNA in here, but I've also got it mixed in with some of that extraction buffer. So I need to do one last chemical reaction. And this chemical reaction is going to pull the DNA away from that extraction buffer. So I'm going to add isopropyl alcohol. This is like the rubbing alcohol that you might buy at the grocery store in the first aid section if you cut yourself and need to clean the cut. This is not alcohol that you want to drink. And I'm going to add that alcohol into my test tube here. And as I added the alcohol, you might notice some stringy pieces of DNA have actually formed up the side of the test tube. All of this right here is DNA. So I'm going to take a glass stirring rod and insert that in there and see if I can grab this big chunk of DNA. My kids always think it looks like a booger, like out of your nose. That right there is DNA from a strawberry. Has anybody ever seen real DNA before?
All the students, please give a big hand, please. All the students are requested to give a big hand. Really amazing. We have never seen actually.
Yalula, voila.
Okay, my next one, I have a little magic trick for you. A little science magic trick. You guys know that magic is usually just chemistry or physics, right? But it can be really fun to pretend it's magic. So I'm going to take this bottle here.
Sorry to interrupt you. Sorry to interrupt you. Here we actually, in this free Zoom slot, we have 9 minutes time is left. Once these 9 minutes come to an end, we have to relog in again. So in this Zoom session, we have only 9 minutes time is left. So once this 9 minutes time is completed or over, we have to, you are requested to relog in again. Okay. And so that we can continue our session. Okay. Yeah, please
Okay.
go ahead, Melissa.
Okay, so you can see that in my jar here, in my plastic bottle, I have some white beads and I have some blue beads. I'm going to shake up this bottle and I want you to watch what happens to the beads. And then I want you to see if you can tell me what's going on inside this bottle. Is everybody ready?
Ready.
Yes.
Well, there, the blue beads all went to the bottom and the white beads all went to the top. But now watch what they're going to do. You see the white beads and the blue beads moving? They're coming back together in the center of the bottle.
Slowly.
So how did I do that with the beads? How did I get them to mix and then get all the white ones to float to the top and all the blue ones to float to the bottom? And then how did I get them to come back together?
It looks like a magic for me.
It's my science magic trick. What's going on here? Anybody have any guesses?
I am not a science teacher. Any students have any guesses about this particular experiment?
No, sir.
No, sir. No, sir. No, sir. It is a good experiment, sir.
No, sir. It is a good experience.
Please go ahead and
No,
explain
sir.
the
It is a
magic.
good experience. It is a magic.
Please go ahead and explain the magic behind it. We are clueless at the moment.
So how many of you guys think that the white beads and the blue beads are made out of the same thing? Are the white beads and the blue beads exactly the same?
Yeah. Question is for everyone. Can anybody guess the answer? Navisai, can you answer to this question?
It turns out they're actually not the same. They look the same. They both look like they're the same kind of bead, but it turns out they're actually made out of 2 different kinds of plastic. And hopefully in one of your science classes, you've talked about the idea of density. Density means how tightly packed the particles in an object are. So something where the particles are really tightly packed together is going to be really dense and it's going to sink. And something where they're not as dense is going to float. So it turns out that the blue beads are really dense and the white beads are not very dense. So when I mix them up and shake them up together, the blue beads want to sink to the bottom and the white beads want to sink to the top. Now the other trick is, it looks like this liquid and this liquid are the same thing because they're both clear liquids. These are not the same liquids. One of these is water and one of these is an alcohol, kind of like this alcohol that we used earlier. So when I shake the bottle up, I mix the 2 of them together. And when they're mixed together, their density is less than the white beads, or sorry, greater than the white beads, but less than the blue beads. So we get white beads, mixed liquid, and blue beads. But these 2 liquids don't really like each other. They don't like to be mixed together. So when I let them just sit there for a minute, they separated. and the alcohol went back to the top, and the water went back to the bottom. And it turns out the water is more dense than the blue beads and the white beads, and the alcohol is less dense than the blue beads and the white beads. So when the liquids are mixed together, they fall in the middle of the two beads. When the liquids separate apart, one of them is less dense and one of them is more dense. So it has to do with the fact that we think the beads are the same and the liquid is the same, but in reality, there's 4 different things in my bottle right now. There's water, dense beads, less dense beads, and alcohol. And so that creates that little magic trick when we mix them together.
So Melissa, actually, what is the principle that works behind this particular experiment? Can you share the particular principle that is working behind this particular experiment briefly?
So the whole reason that this bottle works is because of that idea of density. How dense are things, and that you can't tell density just by looking at something. And I think this bottle is also good because it makes people ask questions. You asked me earlier, how would I come up with a good research question? Well, a good research question starts with something weird or unusual, a phenomenon that you want to know more about. So you watch this bottle work and you go, wow, that's really weird. I wonder what's happening there. And that leads to a really good research question, because then you start studying the scientific idea, the scientific principle, which is density. So yeah, this is a good example of the good beginnings to a research project.
Absolutely, absolutely. We're really thrilled by this experiment, but still we have one more experiment left, I think. Am I right?
Okay, this next one's going to be a little tricky. I'm going to have my daughter help me because my hands are going to get a little bit messy.
Students, don't go away from this session. Actually, we have 3 minutes time left. One more interesting experiment is coming up within few minutes. And here we have in the slide, we have 2 minutes time is left. Once this 2 minutes time is completed, you can re-log in again and to witness another experiment that's going to be very interesting.
I'm going to need you to help control where the camera's at.
Yeah.
Well, don't shut it so much that I can't see what I'm doing. I'll lift stuff up a little bit. Okay, so in the United States, in my state of Idaho, we have an animal called an elk. Do you guys know what an elk is?
I have no idea.
An elk is kind of like a deer.
Yeah.
It's a really big deer. It's a big animal. They have antlers, and they look a lot like a really big deer. So in class this week, we're talking about the body parts of that elk. So what I have here is part of an elk. Oh, that's actually a good idea. And then move it back over there. So what I have here are the body parts of an elk. These right here, and right here are the lungs of an elk. These lungs are made up of kind of a spongy tissue that lets the elk be able to take in oxygen and get rid of carbon dioxide. And then connected to those is a tube here. I don't know if you can see this tube very well. This tube is called the trachea. That's actually this tube right here on us that we breathe in and out of. So this is the elk's trachea, and it connects to these lungs. Then right here in the middle is the elk's heart. The heart of the elk.
Looks like a human heart. Looks like human heart, Melissa.
Yeah. It's actually just slightly bigger than a human heart, but it's about the same size. One of the big differences is these 2 little chambers at the top of the elk's heart. This little chamber right here, it's called the atrium. This atrium is bigger in humans. In the elk, the atria are kind of tiny. They're just tucked right up here on the top. Atria can be kind of tiny because their main job is to hold the blood while it's waiting to get pumped. But you'll notice most of the heart are these two big chambers here called the ventricles. The right ventricle is kind of small. The